Dear colleagues, welcome to our monthly dialogue. First of all, I want to excuse our editor-in-chief, Professor Filippo Crea, because we have some technical difficulties for his connection, but he's listening, so don't worry. He will be able to really respond to questions or anything that comes up. So today, it is my privilege to have here with us Professor Paul Ridker. I mean, everybody knows Professor Paul Ridker, and the title of the dialogue today is Inflammation and Atherosclerosis, moving from CRP to therapeutic targets. And I think we're going to start by introducing the CV of Professor Ridker. He doesn't need that, but nevertheless, I like to do it because he's one of the stars in our field. So he's a graduate from Harvard Medical School, and at the present, he serves at the he has the title of Eugene Braudel Professor of Medicine at the Harvard Medical School. And also he's the director of the Center for Cardiovascular Disease Prevention at the Brigham and Women's Hospital in Boston, Massachusetts. So over the last 30 years, Dr. Ridker has made many advances of cardiovascular medicine. So he has to count in his CV that he and his team were the first to have the proof of principle for the inflammation hypothesis of atherosclerosis in humans. They were the first to demonstrate that statin therapy is both lipid lowering, but also anti-inflammatory. He was the first with his team to have an FDA-approved diagnostic test for vascular inflammation, the high sensitivity CRP. He was also with his team the first to prove that the anti-inflammatory treatment for heart disease could have a successful end with the canakinumab trial. And also, they brought into clinical practice worldwide the concept of residual inflammatory risk that nowadays is in every single Congress we discuss on inflammatory risk. So on multiple occasions, Dr. Ridker's work has altered clinical guidelines for diagnosis, treatment, and prevention of atherosclerotic disease. We are really fond that he is one of the most important cardiologists nowadays. As a direct consequence of his work, multiple novel anti-inflammatory agents targeting interrelated aspects of heart disease, ranging from chronic atherosclerosis to acute ischemia to congestive heart failure, are under development. So he is moving pharmaceutical industry as well, because all these the major pharmaceutical companies are investigating in this area now. So his work, along with work from basic science laboratories and translational colleagues, has helped to move to the clinic the first new target for atherothrombosis in 40 years that is not related to cholesterol, blood pressure, or coagulation. So it's a new area that is open. So he's also known because he has a strong leadership on clinical trials development. So he has been the leader of 15 major multinational randomized clinical trials, including Prevent, Jupiter, SPY1, SPY2, Cantos, CIRT, Prominent, and SIRIUS. So he has been continuously funded by the NIH, and he is the recipient of multiple honorary degrees and international awards. Dr. Ridker is the Distinguished Scientist of the American Heart Association, and he is now an elected member of the National Academy of Science in the USA. So he has an age index of 260, and he has been cited in the literature more than 3,000 times. So he's, if you look into biomedical citation references, he's one of the 15 most cited biomedical researchers in any field worldwide. Therefore, we have here a star. So it's good to have him. And I think I'm going to start with this dialogue, asking him questions that I think will help to our audience to understand the case of this person both in the scientific terms and also as a personal, in the personal area. So indeed, it's a pity that Filippo cannot participate, because he was with me. Very, very fun to have this dialogue. So we have noted that your first studies were on epidemiology and coagulation. So were these your first scientific loves?
So Lena, first of all, thank you for this extremely kind introduction. And let me thank you and Filippo and the European Heart Journal. I'm very honored to be part of this European Heart Journal dialogue today. And I'd also say the timing of this is just superb, because we've just had the first approval by the US FDA of an anti-inflammatory drug for broad use. We've had approval in the US guidelines that you did in the European guidelines a few years ago. So it's very exciting time for us. And I'm also very glad you used the word team multiple times in your introduction, because that is what this is. We all work in teams. And it's nice that I get this recognition today. But the fact is, many, many people have contributed to this work. That's the way this begins. So your question is correct. I started in epidemiology and coagulation. In fact, when I came to the Harvard Medical School in 1981, early 1982, I took a course with Charlie Hennekins and Julie Buehring. They taught an epidemiology and statistics course, introductory course for students. And that course would be very influential for me. So I learned a lot about core epidemiologic concepts from Professor Hennekins. And many of you will recall that he ran the Physicians' Health Study, the aspirin trial, that those of you who take aspirin can thank him for that. And that aspirin is important because my initial interests were, in fact, in understanding why do so many heart attacks and strokes occur in people who don't seem to read the textbook. When you're rounding the coronary care unit, not everybody has risk factors. And the question was, what's going on? What's the unmet risk? It turned out the coagulation really wasn't the answer, but that is where we began with things like factor V Leiden and genetics and thrombosis. But the inflammation shift is what really changed, and I think has dominated our work ever since then.
Yeah. So what attracted to you to inflammation, to this new area? What was the reason you started, that was such a drive in the inflammation area?
Well, you have to remember that the work that we do is translational. So there's basic science going on in the background. That basic science was contributed to by a lot of investigators worldwide, including my colleague, Peter Libby, who would become my chairman of cardiology sometime down the road and a partner in this for many, many years. Other basic scientists were involved who really developed a lot of core issues there. But some of this also came out of clinical work that Filippo knows very well, because Attilio Masri and his group in Rome was talking early on about inflammation as a result of acute ischemia. Now, that's a really important issue, because that's true. Inflammation does follow acute ischemia. But the unknown back in the mid-1990s that really began our contributions to this was to find out, could the inflammation precede the onset of that ischemic event? And that's where the Physicians' Health Study database became so useful, because there were these blood samples from healthy middle-aged men. We would repeat this in the Women's Health Study, where they're healthy middle-aged women who had given us blood samples way before there ever was the onset of ischemic disease. And that changed the paradigm. So while Attilio and the folks in Rome had done an excellent job, including Mark Pepys, among others, describing the fact that ischemia leads to inflammation, we now knew that inflammation precedes the onset of heart attack and stroke, which puts it in the same category as blood pressure and LDL cholesterol. It's a determinant of future risk. And that really is what changed this field, certainly from our perspective. And in fact, Professor Masri wrote an incredibly generous editorial to our New England Journal paper about looking at the dark side of the moon. And I had the opportunity to visit Trieste a few years ago and sit with him. He's a real loss to our community, because he was a real pioneer in this area.
Yes, it's true. Okay, so your initial work focused on CRP, right? Initially CRP, afterwards high sensitive CRP. So if you were to start again, would you have focused on a different inflammatory biomarker, or you would have initiated your research with CRP?
I'm almost loath to admit this, but maybe for younger people, it's a good idea. I wrote a grant to the NIH that did not get funded, where we thought we would measure a whole panel of sophisticated inflammatory biomarkers, ICAM, VCAM, P-selectin, E-selectin, IL-6, IL-1. The grant was not funded. In fact, it got a pretty bad score. It kind of said there's a young, naive cardiologist who thinks you can measure inflammation systemically and learn something. So the high sensitivity CRP was an assay that we helped develop with the help of two clinical chemists, Nader Rafai, a long-term friend, and Russ Tracy of the University of Vermont, to just get pilot data, honestly, for the resubmission of that grant. And of course, that pilot data became the 1997 New England Journal paper that sort of pointed out that you can measure CRP in advance. But I admit, I'm as surprised as anyone that this many years later, we haven't found something better. I think that's partly because maybe we were lucky that picking a downstream hepatic biomarker at the end of this cascade, we now know many years later, NLRP3 to IL-1 to IL-6 to CRP, but at the time that was novel data. And maybe by picking a downstream biomarker is why other blood biomarkers haven't improved upon that. But that's part of the way that science sort of works.
Yeah, exactly. And then you came with the idea that statins were both lipid lowering and also anti-inflammatory drugs. How did you reach this idea just to run a trial? Yeah?
Well, I would recommend again to young people, have terrific, smart colleagues who live right near you. So the story there is an interesting one. Mark Pfeffer lived across the street from me at the time. Mark and Gene Brownwald and Frank Sachs had run the CARE trial. Those of you who remember, this was a pravastatin secondary prevention study, but again, there was biobank available to us. Mark was incredibly generous to let us use that biobank. And so I think we're talking here 1997, 1998, we were able to demonstrate that the statins were both anti-inflammatory in the sense of lowering CRP as well as lipid lowering. That became very important because, as we all know today, statins are really absolutely incredible drugs. They work very fast. There was at the time controversy about why would your risk go down way before lipid levels change, before the plaque characteristics changes. And the vascular biology community was saying, look, there may be these anti-inflammatory effects. So that was a very fortuitous issue at the time for us. And ultimately, that would lead to our Jupiter trial and this observation that we can actually change primary prevention by thinking about rapidly, greatly expanding the number of people who would qualify for statin therapy.
Right. So now let's go back to one of the first trials with to prove the anti-inflammatory hypothesis. So Cantos, why you think that the Cantos data, Cantos results never reached the clinical arena?
So Lena, I'm gonna expand that a little bit. When we did Jupiter, so again, Bob Glynn is the biostatistician, Peter Libby, who was very involved in these early studies with us, we were trying to simply establish that we might want to use CRP to identify high-risk individuals who might benefit from lipid-lowering and anti-inflammatory therapy. So Jupiter comes along, we finish this primary prevention trial, and we have a phenomenal result. I mean, a 44% reduction in major vascular events, a 63% reduction in myocardial infarction. And I would point out, it's the only primary prevention trial study that ever showed a reduction in all-cause mortality. But like many of you on this call, I suspect I see the world through inflammation-colored glasses. And I've always said that maybe the success of Jupiter was because we really exploited the fact that these statins, as lipid-lowering anti-inflammatory drugs, are doing two things. But as Peter would remember well, and others on this call, that was a lipid-lowering trial. To really test the inflammation hypothesis, we would have to pick a drug that hit the specific pathway that we had to guess was crucial to this process. And again, from a historic perspective, Peter and I would get on an airplane. We flew to Basel, spoke to folks at Novartis. To their incredible credit, because canakinumab was being used as an orphan drug for some very rare IL-1 beta overexpression syndromes. I have to be honest and say, I think they barely knew the drug was in their armamentarium, because they were like, you want to use that drug for what? And we said, look, we think this might be the perfect test of the inflammation hypothesis of atherosclerosis, because we know that as a monoclonal antibody, it's going to do one thing, going to inhibit IL-1 beta. By doing that, we should inhibit downstream IL-6. That will in turn lower CRP, which is the biomarker, but not the specific process. So incredible credit to Novartis to allowing us to undertake what you know as Cantos, 10,000 patients, many year-long trial. And I remember well our presentations at the ESC in Barcelona in 2017, where we had back-to-back papers we had to deal with. The one that this audience probably remembers best was the atherosclerosis paper. And that's important because it did prove the hypothesis. Cantos pointed out that if you inhibit this process, the two higher doses of the drug, the 150 and 3 milligram doses of the drug, gave us the same 15 to 17% reduction in MACE that we get from PCSK9 inhibitors. But we didn't change LDL, we didn't change ApoB, we didn't change blood pressure, we didn't change hemoglobin A1C, we just lowered that central inflammatory process. So for the vascular biology community, that was a home run. But what people forget is that we also published the same day. One was a New England Journal paper, one was a Lancet paper, the data on non-small cell lung cancer. And we lowered the risk of non-small cell lung cancer by 45, 50, 60%. So Novartis decided to repurpose canakinumab into an oncology arena. So really, that's the only reason that canakinumab itself did not move into our sphere. And we're very excited about what's going on now with IL-1 and inflammatory inhibitors in the setting of cancer, but that's really a different discussion.
Yes, exactly.
Well,
So-
can you hear me?
Yes, we can.
But we can.
Well, okay. The Wi-Fi now is working much better, so I'm back into the game. And thank you, Paul. I'm noting that we have a massive audience today,
Yes.
the largest ever. So the combination of political and inflammation is magic. It's extremely attractive as expected. Nothing else at the moment. Lina, please continue.
All right. So going back to science. So you already mentioned that colchicine is now an option, right? So in the trials, there were some safety signals. No, what do you think about that?
You know, Lina, let me talk about colchicine, because I think it's a great translation to the clinic that we are all probably underestimating how useful this could really be. So what Cantos provided was proof of principle in a very sophisticated, very expensive, very complex clinical trial with all the bells and whistles that we expect. And we really learned this really, really matters. And the other things that Cantos taught us was the magnitude of inflammation inhibition, the downstream reductions in IL-6, the downstream reductions in CRP. These are the things that determine benefit. And it was beautiful trial in terms of the outcome, but it's a monoclonal antibody and it's being used elsewhere. So the colchicine story is worth talking about. Mark Neidorf, among others, had done an open label study that showed benefit, but it was open label and they need to be repeated. And then two major trials got done. And I have to give credit to the investigators. The two teams, one led by Jean-Claude Tardif, which was the Colcott trial, one led again by Mark and Neidorf and his colleagues, the large-scale Lodoco-2 trial. They were done on a shoestring. These trials did the most important things. They randomized, they double-blinded, and they gave out the drug. But between Colcott and Lodoco-2, we have two major New England Journal papers. Each trial is about 5,000 patients. One is chronic stable atherosclerosis. The other is patients who are roughly 15 to 30 days past a myocardial infarction. So again, pretty stable coronary disease. They show 25 to 35% reductions in risk for MACE and MACE plus.
Right.
Now, think about that. Everybody I'm sure on this call has prescribed drugs like ezetimibe. We all push for PCSK9 inhibition as drugs on top of statins, but those drugs are delivering 6% for ezetimibe, 15% for PCSK9 inhibitors. Now we're talking 25, 30% risk reductions on top of statins. That's a big risk reduction. So personally, I think we should be thinking about colchicine much more aggressively than we are. That being said, Lina, there are some issues with colchicine. For those of you who may not have prescribed it, we don't want to give colchicine to patients with severe chronic kidney disease. We don't want to give colchicine to patients who have severe liver disease. The drug is hepatically and renally excreted. And those patients, it's really a relative contraindication for it. But, you know, a large number of patients on a global basis with chronic stable atherosclerosis would, in fact, qualify for colchicine. I have to admit, those of us in the vascular biology community are very excited to see the US FDA approve colchicine just a few weeks ago for a very broad label. It's actually approved for the use in patients with atherosclerosis or high-risk primary prevention. That's not what I'm doing. I'm using it in very high-risk secondary prevention. But I think that's quite remarkable. And while the European guidelines, the Canadian guidelines put it in a few years ago, the ACC-AHA guidelines just did so recently. One other clinical caveat, Lena, if you're thinking about colchicine, and you should, because it's very effective, I would be, tell your patients certain drugs have interactions. If your patient or the primary care doc happens to put them on a CYP3A4 inhibitor or a P-glycoprotein inhibitor, what I'm really talking about is ketoconazole, fluconazole, maybe cyclosporin. Just stop the colchicine for a week or two. That's pretty routine. But talk about safety, Lena. I think it's interesting. You know, patients with familial Mediterranean fever. Now, I, as an American, never see that. But those of you who work in Israel or Lebanon or Syria or North Africa, tens of thousands of patients take 2 grams of colchicine a day for 30 or 40 years. There's no increase in cancer. There's no increase in outcomes, and they do very well. So familial Mediterranean fever is a model for this, and I think it tells us a lot about safety.
Right. So let's go to the future now, right? So what novel approaches are you investigating? I'm talking about the anti-inflammatory route here, right? So do you have information that in which you can expand now at the present on the future road for inhibiting inflammation?
Well, again, I want to come back to your notion of teamwork, Lena, because one of the things that's been quite extraordinary is to watch these very large clinical trials develop in the anti-inflammatory space. And we have investigators globally who contribute enormously to this. I think I see Raul Santos on there. I see Wolfgang Koenig on there. This is really kind of fun for me today to see so many friends. Every major pharmaceutical company right now is developing targeted anti-inflammatory therapy. I think many of us think that combined lipid lowering and inflammation inhibition could well be a pretty standard intervention down the road. We were working with a small company in Waltham, Massachusetts called Corvidia on an IL-6 inhibitor. That drug you know as ziltivekimab. So this is an IL-6 ligand inhibitor. And Novo Nordisk, again, deserves enormous credit for thinking about moving out of the GLP-1 and diabetes space and has funded 3 ongoing major clinical trials. One very important one is called Zeus, because that's the patients with chronic kidney disease who I don't think we can give colchicine to. Zeus will be 6,000 patients with known atherosclerosis, elevated CRP, so they have residual inflammatory risk, all on a statin, getting either IL-6 inhibition or placebo. It's about halfway enrolled already. There's a second trial called Hermes that has been launched that is in heart failure with preserved ejection fraction. It's a very controversial arena to enter the heart failure world with an anti-inflammatory, so it's a great experiment to see whether that works. And there's a third that's just been discussed publicly called Artemis, which is going to be an acute coronary ischemia trial, which goes all the way back to the work from Attilio Masri and his colleagues looking at acute ischemia in that setting. So, and those are just the ones that I think are publicly announced. I think there's many other things going on, and there's other pathways that are being explored. So it's a very exciting time. Yeah.
So from your conversation till now, you don't think that we can have any anti-inflammatory safe?
Lina, sorry, can I interrupt you?
Yes, please go. Well, we cannot hear you, Filippo.
Lina,
yeah.
Can you hear me? No, okay, practice. And thank you, Paul, for mentioning my mentor, Attilio Masri, who's been a giant in the field of inflammation. You are another giant in the field of inflammation, but in the audience, I see a third giant in the field, Peter Libby. Peter, I'm sure that we have questions for us. Dialogues are very interactive, so I also invite all of you to ask questions. This is really a great opportunity to ask questions to Paul Whitaker, to Peter.
Well, Filippo, you're breaking up on me, but it's a delight to see Peter here because Peter has been a
questions.
Okay.
Yeah, so Filippo, you're breaking up on me, but Peter has been a partner in this for almost the entire journey, honestly. And he and I work together very well, in part because our skills are quite different. You know, Peter really knows the bench vascular biology territory in a way that I can read about, but I don't do in a daily practice. And I, who sort of live in the clinical trial translational world where Peter reads about but doesn't do daily. So it's a great team that we've had. That's why Lena's original thing about teamwork makes me so happy. But yeah.
Yeah. So Filippo, we lost you, but nevertheless, for everybody, if you have any question, please write it in the chat, and we'll try to have Paul responding. So I'm going to continue because Paul, you are so fond of inflammation, but with all these possibilities of blocking inflammatory pathway, so you don't anticipate any relevant safety issue, right?
Oh, no. Safety is going to be an issue with any class of drugs. And you know, when you tamper with inflammation inhibition on a chronic basis, you are likely to get some small increases in infection. We saw that in Cantos, but it's very important to point this out. It was staph and strep. It was not opportunistic infections. It was not reactivation of TB. It was not HIV issues. And bringing patients in a little bit earlier with antibiotics is a good idea. Remember that our rheumatology colleagues and our gastroenterology colleagues, they've been using IL-6 inhibitors for 20 years, and it's been very safe in their hands. They've just learned, as the cardiology community is going to have to learn, if someone has an infection on these drugs, get them in a little bit earlier. Don't just assume it's viral. Give them some antibiotics, and I think we're going to be fine. But we have to learn the lessons that our rheumatology and gastroenterology colleagues have, because, you know, we have patients with inflammatory bowel disease or other rheumatologic disorders who are on these drugs already on a chronic basis. So we know it can be done.
Right. So we have 3 questions here so far. Dr. Daniel Di Nano is asking you if you propose to use CRP measurements to select candidates for colchicine treatment.
It's actually an excellent question because the trials did not do that, but I do. And let me tell you why. I think that we want to target anti-inflammatory therapy to the patients most likely to benefit from it, and those are the ones who have this concept of residual inflammatory risk. It's a little bit like LDL. I could give LDL lowering drugs to everyone, but I do measure LDL to figure out I think it's elevated. Blood pressure, I measure blood pressure in everyone because I want to make sure I'm targeting things well. So I do measure CRP, and we've learned from our colleagues at Ledoco and Colcott that even though they didn't measure it on everybody, when they had the measures, the CRP levels were 3.5 to 4 in those trials at baseline. So I think that's the way to go. It's also important because we're in a new era. We can't give everybody every drug. We lucked out with statins, and we probably lucked out with some anticoagulants and aspirin. But we're in an era now where some people are going to get perhaps an Lp(a) lowering drug. Some people might get a diabetes drug. Some people might get this. We've got to figure out who drugs work best for. So yes, I do measure CRP routinely, and those are the ones who, if it's elevated after being on a statin, that I do treat. All
right. So there are a few more questions here. Maybe there is one from Enrique Sanchez, who is asking if colchicine can help in diabetes microangiopathy. You think can help patients with diabetic microangiopathy?
Well, we don't know. Colchicine has been tested for chronic atherosclerosis. There's people have proposed studies for metabolic syndrome and other issues, but right now that's the data we have, is it works for the disease that's most going to kill the most humans on the planet. So it's important.
All right. So there is a question here on siltiberkimab, and this is Dr. Anna Tomaszewska-Kasberuk. Sorry if I don't pronounce your name well. So Anna is asking, what you do when a patient's treated with the antibody gets sepsis?
So again, let's not talk about ziltivekimab. Let me talk about the class of IL-6 inhibitors, which includes tocilizumab and many secukinumab, many other IL-6 inhibitors. In standard rheumatology and GI practice, it's difficult to use CRP as your measure of infection because it'll be artificially reduced by the drug. So the SED rate becomes very useful in that setting. But again, I want to point out that most patients who are on these drugs chronically do quite well. And our gastroenterology colleagues and our rheumatology colleagues simply have learned, if they see the signs and symptoms of infection, a temperature that goes up, a SED rate that goes up, temporarily stop the drug, initiate antibiotics a little earlier than you might otherwise. And patients have done very well. This is not a new class of drugs.
Right. Well, Peter Levy is with us. Do you want to ask anything or add a comment here? You are muted. Now.
Yeah, okay, I'm sorry, I was muted by the host. Well, I think the theme of teamwork has come up, and Paul and you both articulated it. You know, I once heard Brown and Goldstein give a talk on the 10th anniversary of the recognition in Stockholm in the Golden Hall of Ratshuset in Stockholm. And the theme was partnership. And they talked about Gilbert and Sullivan and Rogers and Hammerstein. And it's been, Paul, I hate to say this, but it's been 30 years that we've been working together. And Paul articulated it. It's the interlocking skill sets that really have helped to move things along. And we've gone shoulder to shoulder in trying to get sponsorship of trials such as Jupiter and Cantos. And it's been a great journey. I've learned a great deal. Everything that I know that's true about clinical trials, I learned from Paul.
Right. That's very good. All right. So a new question regarding these future options that we may have, right? So what is your opinion on the possible relations between atherosclerosis and CHIP? You know, this new test that is being really, well, it's different groups are trying to investigate on this. Do you foresee clinical implications in that side? I mean, we will be able to.
Sure. So just to be clear for everybody, so CHIP is clonal hematopoiesis of indeterminate potential. This is an area where colleagues of ours, Sid Jeswal, Ben Ebert, Peter's been very involved in this. Some very exciting papers were published a few years ago about this being a acquired genetic risk, right? This is something that you're not born with, but as you age. Exactly. And I think for me, the really interesting part of this has been to bring hematology and bone marrow function back center stage into atherosclerosis. Lots of work done by many people on this call, I suspect, about neutrophil function and the way the bone marrow works. There are companies now developing CHIP assays, so it's not just something that can be done in a unique setting. We have to figure out, as clinical translational scientists, where might the sweet spot be? Are there ways to intervene on patients with CHIP? I think you're asking this because within Cantos, we did show that IL-1 beta inhibition was somewhat better in terms of risk reduction among those who did have CHIP, but it also worked among people who didn't have CHIP. I'm excited about it. I'm looking forward to it. We'll see where the field goes.
Exactly. Okay, and what about fat? Perivascular and ectopic fat. What's the contribution of that fat to the heart in different functions of the heart?
Sure. So two things about fat. The first is we should all exercise, diet, stop smoking, and primary prevention really matters. All those things that we talk about matter for lowering inflammation. They all matter for vascular risk. You're really asking, though, about perivascular fat, which is an issue about fat surrounding the coronary arteries. There's a novel technique, the fat attenuation index, that has been promoted for measuring that. It's a little bit like CHIP right now. It's an interesting issue. There's ways of doing this. We need some clinical data to convince us that it is useful. Even if it's real, the question becomes, is it useful? And so there are some studies being done now to find out whether things like FAI and others might matter here. It is also true biologically that, you know, fat cells in general are in some ways a virtual production factory for cytokines. So again, avoidance of fat's probably a pretty good idea.
Good. So now I'm going to ask you something that Filippo is interested in, and he was telling me. So what about patients with low levels of LDL and also low CRP levels? So how could you address residual risk in this population?
Lena, the sole fact that you're asking this question makes me smile. If everybody simultaneously measured hsCRP and LDL, we would have different groups of patients. We'd have some individuals with high CRPs and high LDLs. We recently published data in the Lancet from a combination of work from the prominent reduce it and strength trials showing these are extremely high-risk individuals. We showed that people with well-controlled LDL, but an elevated CRP have risks that are almost just as high. And we had the lowest risk being in the group you just talked about, the low LDL, low CRP. But we only know you're in that group if you actually make measurements of both of these things. So that's number one. But number two is there are still events occurring in patients with low LDLs and low CRPs. So again, for the young people listening, there's a lot of room here to figure out some new ideas. There are events going on that might be due to unique circumstances that are not picked up either by the lipid piece or the inflammation piece, or that are related to lipids and inflammation that just aren't picked up as well by LDL per se or CRP per se. So you may need more sophisticated lipid measures or more sophisticated inflammatory measures. It's a ripe arena for future work.
Right, and here comes the issue. If we do need more specific imaging markers or markers for the plaque characteristics, erosion versus a fissure. So these are different aspects that maybe we don't have completely understood as well. No, we don't have biomarkers for this. And imaging is improving a lot, and I think that in the future will be even better. Right? What's your opinion on that? The complementarity of biochemical biomarkers with imaging.
So Lena, I think you're asking a very important issue. First, the natural history of acute myocardial infarction has shifted during our careers. I remember when ST elevation MI was very common. Now our house staff get excited when they see it. Plaque erosion has now become much more common, and rupture seems to be less. And these are issues that affect, I think, the changing demographics of the disease, who's getting the disease at what age, and the fact that many patients are on aspirin, they're on statins. And so the actual way the disease presents is also different. So yes, we need to have some new biology. We also wouldn't it be great to have an imaging technique that could tell us, aha, this patient has preferentially an inflammatory problem, or aha, this patient preferentially has a coagulation problem. Those kinds of imaging tests are very sophisticated. It's usually tying a biochemical parameter to the actual imaging test. Peter and I have a colleague, Ralph Weislater, across town at Mass General, who's been a pioneer in this area, trying to link biologic markers to imaging. I think that's an exciting arena. But look, just corneal calcium scanning clearly is a very good way of knowing you've got a high-risk patient. And the question is, can we figure out, using some novel imaging techniques, something that'll really help us about the inflammatory piece? We're not there yet, but I sure hope we find something that allows us to say, aha, this patient will really benefit from this kind of therapy, and that patient from a different kind of therapy. Right,
and Filippo also wonders why high sensitivity CRP and the concept of residual inflammatory risk has been stronger in the US than in Europe. Do you have any idea why? It's because you are there.
No, Lena, I'm smiling because the very first time the word residual inflammatory risk appears in the literature is in an editorial commentary that I wrote in the European Heart Journal, actually. So I'm very grateful that the European Heart Journal, who's supporting this dialogue today, was the place that appeared. That was partly to address this issue. I think some of this also is that in Europe, the SED rate has been used to traditionally measure inflammation, not so much CRP like we have. But look, a lot of the basic original work also came, again, from Rome, from London, about CRP some time ago. I think we're all getting there. I think at the end of the day, some of this has to do with marketing and promotion, perhaps. Again, this is going to be a challenge for colchicine. We won't see any marketing for this, right? So let me give you another example, Lena. There's a lot of measurement of Lp(a) going on. Now, I measure Lp(a) in my patients. Again, measure it once. It's a genetic marker. Don't need to measure it twice. But there's been an epidemic of Lp(a) measurement, I think because we're anticipating we may see some drugs that do this. But, we don't know yet. Yeah. I'd also point out that, our guidelines have been telling us to measure triglycerides and HDL for 40 years. But we lack data saying that raising HDL necessarily lowers risk, or lowering triglycerides necessarily lowers risk. We have evidence that lowering inflammation lowers risk. So maybe we'll see more screening in the future.
All right. So there are some questions here in the chat. One is from Dr. Wayne Kissmeyer, and he's bringing into the discussion the bempedoic acid. So what does bempedoic acid does? It does on CRP, and maybe the combination of a statin with bempedoic acid for high-risk patients may work. What is your opinion of this new drug in the scenario?
Well, so Steve Nissen and his colleagues recently showed in the CLEAR Outcomes trial that bempedoic acid as monotherapy among patients who were either truly statin intolerant or just didn't want to take a statin lowered risk by 12, 13%. That's a good finding. It's a new agent, therefore, for patients who truly can't take a statin. It's not as large a benefit as we saw with statin therapy. I give the CLEAR Outcomes investigators great credit. They enrolled a very large number of women, which is really important. And the women benefited just as much. So I think that was also a very good thing they did. We did some sub-studies, as did others, that showed that yes, bempedoic acid also lowers CRP, as do statins. So presumably there's some commonality mechanism there. But again, I think that what's interesting to me is that at least in US practice, when we give bempedoic acid, we typically give it in combination with ezetimibe because we're trying to get LDL lowering. But whether or not the anti-inflammatory effects are a crucial part of that or not, we don't really know. We do know there's an event reduction, and I think that's the important issue. But I have to say, Lena, in my practice, maybe because I'm very persuasive, I don't know, I get almost all my patients on a statin. I'm willing to go every other day. I'm willing to go 5 milligrams of rosuvastatin every third day. And I say to them, look, you're taking 6, 7, 8 medications. This is the one that really, really matters. Another thing I do just for this is I never prescribe exercise the same day I prescribe a statin, because they're going to go to the gym for the first time in years. They're going to lift some weights. They're going to get muscle aches. They're going to blame the drug. And so you have to be very careful how we do this. So I believe statin intolerance is real for some people, but it's not as common as the internet would like us to believe.
Exactly. So there is one question here on primary prevention on population cohorts. This comes from Mayra Alejandra Vera Montoya, and she's asking if you consider that CRP should be used to measure inflammation in this primary population cohorts, and to know if they have some problem with inflammation, and established anti-inflammatory preventive therapies in these groups.
Well, so that when we began our conversation, I was talking about work from Charlie Hennekins and Julie Buring, the folks who set up these studies called the Physicians' Health Study and the Women's Health Studies. So cohort data has been available for 30 years, telling us that if you know CRP, you do very well. The MESA study has shown this. Wolfgang Koenig, I think, is on the line. His studies have shown this around the globe, as have the more recent UK Biobank studies. So we know that CRP is a very good marker for this low-grade systemic inflammation. The question is, do we do it clinically?
Exactly. So there is another question from Dennis Clement. This is more on the research area. So he's questioning if you know if inflammation is active in the different areas with atherosclerosis. So different arteries, carotids, coronaries, peripheral artery disease is associated with atherosclerosis at the same level?
Yes, I think the answer to that is pretty well known. We have excellent data for stroke. Stroke reduction was also seen very clearly in both Cantos and in Colchicine trials. CRP is a very powerful marker, as is IL-6, a very powerful marker for risk of all-cause mortality. Peripheral arterial disease is a challenge because while the risk markers work, the drugs have been less impressive to date. And I think we all have to find some drugs that work for very large vessel disease. Small vessel disease is trickier. There are relationships, but it's hard to know about the causality of it because the small vessel disease may well be different biology and a very different set of drugs for, microangiopathy and diabetic eye disease and the like. But for generalized atherosclerosis and aortic aneurysm, yes, is across the board.
Right. So there is one question from Martin Ellis regarding homocysteine, because it was very fashionable years ago, and he's questioning if it's still a relevant target for primary and secondary prevention.
I have to admit, I have not measured homocysteine in a long time, and we don't have evidence that reducing homocysteine reduces risk. So I think the answer there is probably not.
Right. Okay. So I think that we are going to start finishing because we have to stop at the hour. But I think that there is another question coming in. What about fibrinogen? What is your opinion on fibrinogen?
So actually, fibrinogen is a risk marker of the acute phase response as well as CRP. It certainly is a coagulation product that's important. And people have shown D-dimer and fibrin split products also are associated with events. But modulating fibrinogen has proven quite complicated. And so again, I think that biomarkers like CRP and probably IL-6 are the ones that most people are going to be using on a regular basis.
Right. Okay, so let me get to the area of your changing topics. So you have been working for many years in one of the most important medical institutions in the world, right? Harvard. So what brought you there?
Luck. Look, I think that I have been extremely fortunate in my career. I came to the Harvard Medical School with no medical background in my family. I had been an English major in college and I wasn't all that sure if I belonged in medical school. I found research to be extraordinarily interesting. I've also been incredibly blessed with leadership and mentorship over the years. The chiefs of cardiology I've served under, Tom Smith, Peter Libby, who's here today, John Keeney currently, the chairman of medicine I've served under. I mean, it's a list of the who's who in the cardiology world. It's Eugene Braunwald, it's Victor Zhao, it's Mark Pfeffer, it's Joe Lascalzo. You know, if you're blessed to be at a center like the Brigham and Women's Hospital and the Harvard Medical School, where that's the leadership you have for your career, I consider yourself a pretty lucky person, and I fit in that group.
Right. And obviously you already mentioned Professor Braunwald, and really everybody believes that he's the father of cardiology presently. So when did you meet him for the first time? And what has been his impact in your life? How you can analyze this human relationship?
Gene Braunwald is my academic father in many, many ways. I met him when I was a medical intern at the Brigham Women's Hospital. At the time, he was quite intimidating, but I would learn over the years that he's actually a remarkably warm and thoughtful person who really cares enormously about the people who work for him and around him. His contributions to medicine in general and in cardiology in particular are obviously unparalleled. At the end of your internship year, Professor Braunwald meets with each one of us. I thought I was going to be an infectious disease physician. That's what I've been interested in previously. I sat in his office. He called up Tom Smith on the phone and I said, I'm going to send this young man down to you because I want him to be a cardiologist. And that's literally what happened. And to this day, I've been grateful to him for that. Gene has been a father figure to many, many, many of us. It's not just his presence, his textbook, his influence, his way of sort of caring about people. It is just extraordinary. And Lena, we're all incredibly lucky to have him and to continue to have him. He's just as active as he ever has been. And many people on this call, I suspect, get phone calls from him once in a while asking a very specific scientific fact. I laugh about this. Sometimes I'll get a call from Gene's office and he wants something really sophisticated, really quick. And it's always a brilliant question that makes me think. And I go, how does he know more about my field than I do? But that's the way he is. And anyway, yeah.
Well, and I'd like to ask you something personal, because you have an artistic side in your family. Would you like to comment on that?
Oh dear. Yeah, so Lena knows that I have two children who avoided medicine completely. They're both professional writers. My daughter writes for television. My son is a novelist. There's a very famous saying that he brought to my attention that when a novelist is born, the family dies. That's because they tend to write about not their families, but their families. Some people think these books are, some people forget these books are actually fiction. But anyway, for those, I can see Peter Libby laughing. For those of you who read, Andrew has just published a book that does have a cardiologist who works at the Brigham who does inflammation research, but it's not me. Trust me, it's not me. And the stories that he tells are just very funny. But thank you, Lena, for that.
All right. I am reading here that Roxana Miran is thanking you. Are you here, Roxana? You want to say something? You are mute.
I am. The host has to unmute me, please. Yeah, that's great. First of all, Paul, Peter, Dr. Brunwald, Filippo, and of course, Professor Mazzari. The work of inflammation is seminal in atherosclerosis, and we're understanding it better and better because of you, because of the work that you all have done. And I can tell you, and I know you don't like this, Paul, but I measure CRP on everyone. I believe it's like a vital sign, just like I measure blood pressure, pulse, everything else. I believe that as a cardiologist, you really want to know. Just the other day, I just want to give a retrospective kind of an anecdotal thing. I measured, had a woman in my office, and of course I measure LP(a) in everyone. I've done that for years because of Peter Harpel, who comes from this institution at Mount Sinai. And I was convinced that LP(a) had a lot to do, since I looked under the microscope and saw the atherectomy specimens of STEMI patients and non-STEMI patients filled with LP(a). But be that as it may, I just saw a patient and her CRP was 12. I was worried about her. I had nothing else to go on, but I was worried about her. And then 2 days later, I was called that she's in a doctor's office with a atrial fibrillation going at 140 beats per minute. And 220 beats per minute, sorry. And of course she went to the ER, and now other things are going on. And I had nothing else except that. And I wanted to call her. I had her on my list to give her a call to see how she was doing. Just so you know, I mean, this is the work that you all have done that is helping patients. You don't know how many lives you've touched. And I think it will be very, very relevant when we start to see more of these clinical trials. I'm lucky enough to be co-chairing the Artemis trial, but I believe that the trials coming forward, focusing on the work that you have done for decades, are going to be seminal in hopefully reducing the burden of cardiovascular disease and understanding better the role of inflammation in all of these syndromes that we are seeing, and cardiovascular death and morbidity mortalities. But thank you for everything you've done.
Well, Roxana, first of all, thank you for many things. When the core interventional cardiology community is measuring CRP in everyone, it does make me smile. So that's great. I want to say one last thing, Lena, if I can, as we sort of close this down. I do see younger faces on the screen, and I'm very excited about that. What they may not know is that they become the leaders awfully quick. And those of us who are lucky enough to have this academic clinical career feel very blessed to do so. But Lena, your daughter Guiomar is already part of our ongoing clinical trials, and it's been an absolute pleasure to get to know her. And so thank you for helping to produce and train part of that next generation for us.
Thank you very much. Thank you very much.
Lena, can you hear me?
Yes. Go ahead.
Well, I see in the audience another top scientist, young top scientist, Viviana Tacchetti. She's not only a top scientist, but also chairman of the publications committee of the American College of Cardiology. Viviana, you're very welcome. And again, from Brigham. Any comments, questions for Paul?
Well, thank you very much, Filippo, and thank you, everyone. I see a lot of colleagues and friends from over the years, and just to say just how inspirational it is to work with all of you. I think you've all touched the way I think about science and patient care in transformative ways that is really informing how I craft, I think, my future career paths. And so thank you for the opportunity to participate in talks like these. I think they're incredibly inspirational for all of us, and especially the next generations.
Okay. Well, thank you very much, Viviane. So with this, I think we have to close the session. We are really happy to have had you, Paul, with us in these dialogues. And well, maybe Filippo, you can talk. For those that have connected late, Filippo had technical troubles. So that's why he kept his participation to a low level. But he's the person behind all this movement that we have at the European Heart Journal. So if you want to say something, maybe you can. I mean, you're not frozen there.
Well, I hope, can you hear me?
Yes.
Well, I must admit that I'm not in my office, but in a small island close to Sardinia for my short holidays. But I couldn't miss this important dialogue today. I hoped Wi-Fi might have been better, but anyways, I've been with you all the time. It's been a wonderful dialogue. Thank you, Paul, for really joining us for this dialogue. I wish to thank also our colleagues with so many, really so many questions. Sorry, we couldn't address all your questions, but they're really numerous. And this certifies the importance of this dialogue with Paul Ridker on inflammation and other really very, very important colleagues in the audience. I think that the only last announcement I'd like to make is that next month in August, we will not have a dialogue, but you are all invited to a very special editorial board meeting of the European Heart Journal, during which we will celebrate a very successful year. The editorial board meeting will be in Amsterdam on Saturday, 26 at 19:30 Central European time. So will be another interesting occasion to meet together. And I really hope to join physically or virtually all of you for that occasion. So please take note, Saturday, 26 August at 19:30 Central European time. And thank you not only to Paul, but also to Lina for handling this dialogue with these technical complexities. It's been really a very intense and pleasant hours, and another marker of success, again, is the very high numbers of questions we have received. So, Paul, let's hope that we will master inflammation in the future in the interest of our patients. But what we have done, you, Peter, Attilio Maseri, and many others, but the three of you in particular is astonishing. When Attilio Maseri published in 1994 the seminal paper on CRT, the emphasis was on lipids, thrombosis, risk factors, but inflammation and inflammatory cells were not on the scene, on the clinical scene. In the meantime, Peter was preparing the background work, which explained why inflammation was bound to be so important in clinical practice. So this experimental and translational background and the intuition by Attilio and the work you've been carrying out in the following 30 years are the basis for this successful story. Now, inflammation is there to be treated in the best possible way. And what we've shown in your pipeline is really extremely exciting. So thank you, all of you, for the participation. Not forget Saturday, 26, 1930, for what I promise will be a very interesting editorial board meeting. Thank you very much.
Filippo, let me perhaps thank you extraordinarily and Lina for the honor of this dialogue today. But you, Filippo, taking the helm of the European Heart Journal, we know it's an enormous amount of work, but it's work that really matters. So thank you for all the efforts you've made, and thank you for your friendship all these many, many years. It's greatly appreciated.
Well,
thank you, Paul, for your words.
Thank you to everybody who has connected. Bye-bye.